Hi, my name is Anne Kantarjith, and I'm a graduate student in Professor Wei-Shu Hu's lab. Today I'll be sharing with you an example of transcriptome analysis in mammalian cell culture. We can think of a number of applications for microarrays in cell culture, and today I'd like to share with you an example where we used them to study hyperproductivity. Now we can approach this problem from two different angles. One can be to look at different production cell lines, for instance comparing high and low producers, and the other is to look at different culture conditions which are known to increase productivity. So in today's example, we compared three different productivity enhancing conditions in CHO cells. The first was the use of a temperature shift. The second was the combined use of temperature shift with sodium butyrate treatment. And finally, the last condition was the use of methotrexate for DHFR amplification. We looked at the intersect of the results from each of these three studies in an effort to gain further understanding of the trait of hyperproductivity. So if we look at the number of genes we identified as differentially expressed in each of these three studies, you can see that that number is actually quite small. However, if we think of what makes a cell a high producer, we can think of productivity as a pyramid, where a number of functional classes exist at the bottom, and then different changes within each of these functional classes can lead to an equivalent increase in productivity. So really the question then becomes, what are the functional classes which are favorable for high productivity? So in order to identify those functional classes, we use two different kinds of functional enrichment analysis. The first was gene class testing, and the second, gene set enrichment analysis. And this table shows the results of those analyses. If a block is colored, it means that that functional class was identified as significantly enriched within that particular study. So you can see that a number of functional classes begin to emerge as common amongst each of the three productivity enhancing conditions. Among these, we found cell cycle to be reoccurring in each of the three conditions, as well as vesicle-mediated transport. Now, armed with this knowledge, we can think of a way where microarrays could be used as a screening tool in the process of cell line development. If we can use transcriptome analysis to screen and identify high-producing clones earlier in the cell line development process, then we can shorten the timelines to go from transfection to production cell line.